Lesson 7. Question number 1. How does bioremediation work? Bioremediation occurs when microbes come in contact with contaminants such as oil, gas, or diesel in a suitable environment. Question number 2. What is the difference between in situ and ex situ? In situ refers to the original location, whereas the ex situ refers to the off-site.
Hello. So, for lesson 7, the number 3 states questions about what are the advantages and disadvantages of bioremediation. So, the advantages and disadvantages of bioremediation is that one of the disadvantages is that every organic and inorganic pollutant can be remediated. Ex situ control of website organic pollutants may be difficult, and lack of clean site standards, highly permeable soil is required. Then environmental application of DNA are still restricted. So that is the disadvantages. So in here we're going to discuss the advantages of it. So here it goes. So one of the advantages is that it uses microbiologically naturally ability to extract chemicals from water, soil, and sediment using sunlight. So regulation and public acceptance, as well as low implementation cost. Then no need for post-processing, as contaminants are simply separated. So cheaper than other physiochemical methods. And no need for post-processing, as contaminants are simply separated. So cheaper than other physiochemical methods. DNA can aid in the development of multiple degradative pathways for pollutant degradation. So that's all for number 3. So let's continue with the advantages. So it states here that no need for post-processing, as contaminants are simply separated. Then next is it is cheaper than other physiochemical methods. Then the last one, hello. So before we proceed to lesson 8, let's go with the lesson 7, number 4, last but not the least. So where the question is all about, and it is being asked, what are some of the applications of bioremediations? So in here it says that removal of contaminants, pollutants, and toxins from soil, water, and other environments. So some various applications of bioremediation are soils, sediments, and sludges as solids, groundwater, surface water, and industrial wastewater as liquids, industrial air emission as gases, saturated and vadose zones as subsurface environments. So that's all, and thank you for watching. So let's proceed to lesson 8. Hello everyone. So we're in lesson 8, in which I'm going to discuss number 1 and 2, in which the question in number 1 is that what is plant tissue culture? So plant tissue culture is the process of creating more plants from one plant, or plant tissue culture is defined as culturing plants, seeds, organs, explants, tissues, galls, or protoplast on a chemically defined synthetic nutrient media under sterile and controlled conditions of light, temperature, and humidity. Hello, so this is lesson 8, number 2, in which the questions is, what are the advantages of plant tissue culture? So it says that the new plantlets can be grown in a short amount of time. Second, only a small amount of initial plant tissue is required. The new plantlets and plants are more likely to be free of viruses and diseases. The process is not dependent on the seasons and can be done throughout the year. You need only a relatively small space to perform the process, 10 times the plants in 1/10 of the space. So that's where my part in discussing it. So I'll give it to my group mate, Mr. Ubag.
Good day everyone, I am John Rhys Obag. So, I am going to explain our answers in question 1, 2, and 3. So, question 1. What is the role of photosynthesis in plants? Photosynthesis is to allow plants to make their own food by converting light energy from the sun into chemical energy. 2. Describe the structure of chlorophyll. The central chemical structure of chlorophyll has a magnesium atom. 4 nitrogen atoms are found around the magnesium. A hydrocarbon end is also seen. There is a pyrrole ring structure with 4 carbons and nitrogen. The 4 pyrrole rings with nitrogen and magnesium are together termed the porphyrin head. The hydrocarbon end is the pytol tail. 3. Cite the importance of chlorophyll. So, chlorophyll is vital for photosynthesis, which allows plants to absorb energy from light. Also sustains plant life and produces oxygen for the entire planet. Lesson 6, question number 3. Summarize the process of light reactions and Calvin cycle. So, the Calvin cycle, which takes place in the stroma, uses derived from these compounds to make Ga3P or glyceraldehyde triphosphate from CO2 or carbon dioxide. Light reactions, which takes place in the thylakoid membrane, use light energy to make ATP or adenosine triphosphate and NADPH or nicotinamide adenine dinucleotide phosphate. Question number 4. What are all the byproducts of light-dependent and light-independent reactions? So, the products of light-independent reactions and can survive for hundreds of millions of years, whereas light-dependent reactions have lifespans in the range of millions of